Hi everyone, welcome back to my single-cell scATAC-seq video tutorials. In my last video, we performed PBMC data pre-processing, quality control analysis, data normalization, dimensional reduction, and cell clustering. And we got 15 cell clusters from our analysis. In single-cell RNA sequencing data analysis, cells are clustered based on their gene expression profile. We can name the cell clusters using surrogate function finder markers. But in single-cell scATAC-seq data, cells are clustered based on the peaks of open chromatin, and some peaks only contains non-coding genomic regions, so it is more challenging to annotate and interpret cell clusters in single-cell scATAC sequencing data. The best way to name cell clusters is to integrate single-cell scATAC-seq data with an analyzed single RNA sequencing data, which I will show you in my next video tutorial. If you don't have an analyzed single-cell RNA sequencing data, then we can try to name the cell clusters based on the prediction of gene activities in open chromatin. So today I'm going to show you how to name cell clusters using predicted gene activity. Because the open chromatin contains genes that could be transcribed into RNAs, so we can quantify the gene activity for each cell and create a new gene activity assay derived from the single-cell scATAC-seq data. Gene activity from the data could be calculated by the gene activities function in scATAC package. It takes several minutes to run the function, so let's run it now to create a gene activity matrix. So you can see it takes more than 7 minutes to create the gene activities. In this window, you can see we have a data set named the gene activities. It is a large DG matrix data set. So now we can use the counts of gene activities to create a assay object as predicted RNA. Then we can add the predicted RNA assay data into our PBMC surrogate object as RNA assay. Before we do this, we can have a look at the PBMC. You can see at the moment, PBMC only has one assay. It is peaks for single-cell scATAC-seq. So now we can go ahead and add the RNA assay into PBMC object. We can have a look at the PBMC again. Now you can see we have two assays in the PBMC object, the peaks and the RNA. So we just need to normalize the RNA assay data before we see the gene activities in the cell clusters. So we will use the same function for surrogate single-cell RNA analysis. To normalize the data, method will be log normalize. So let's normalize the data. So because we have two assays, we can have a look at the default assay for PBMC. You can see at the moment it is PIX. Then we can change the default assay to RNA. We can have a look again. You can see now it is RNA. So if you are familiar with the throughout PBMC single-cell RNA tutorials, you will recognize some mark genes for different population immune cells. Now we have predicted RNA data from open chromatin, so we can have a look at those mark gene activities using the feature plot function. So we know CD14, LYJ, TREM1, and MS4A7 are mark genes for monocyte. So we can run this feature plot to see which cell clusters are labeled by monocyte mark genes. Let's run. We can zoom in. You can see CD14, LYJ, TREM1, and MS4A7 label cell clusters 0, 4, and 9, so they are monocyte. So we can use other gene activities for mark genes B cells and NK cells. MS4A1 and CD79A are mark genes for B cells. NKG7 and GNLY are mark genes for NK cells. So we can use the feature plot function again to label B cells and NK cells. So let's zoom in again. You can see MS4A1 and CD79A label cluster 8 and 10. So we know 8 and 10 are B cells. NKG7 and GNLY label cluster 7 and 12. So cells in cluster 7 and 12 are NK cells. So we have cell clusters 1, 2, 3, 5, and 6. We know they are T cells. We can use T cell markers such as CD4 and CD8A, AL7R, and CCR7 to label T cells. Once again, we can run the feature plot. Let's see it in a bigger window. You can see CD4 marks cluster 1 and 3. CD8A marks cluster 2, 5, and 6. IL7R and CCR7, they are receptors. Label all the T cell clusters. So as demonstrated by today's video tutorial, we can use gene activities to label cell clusters in single-cell scATAC-seq dataset. In my next video, I'm going to show you how to integrate single-cell scATAC data with analyzed single-cell RNA dataset. See you in my next video. Please don't forget to subscribe my channel.